Hello, I'm Dr. Stephen Pike from the University of Michigan, and I had the opportunity at the World Federation of Hemophilia Virtual Summit to present on behalf of my co-authors on the recent progress on the development of AMT 061, which is etranacogene dezaparvovec, a gene therapy treatment for persons with severe or moderately severe hemophilia B. Etranacogene dezaparvovec is an investigational treatment that uses an AAV5 serotype to facilitate gene transfer to the liver. This encodes the factor IX gene that includes a point mutation, the so-called Padua variant, which makes a hyperactive factor IX. This is actually an enhanced version of a previously evaluated gene therapy treatment called AMT 060, which already has demonstrated efficacy and safety in a phase 1/2 trial. And in that trial, what was observed was stable expression of wild-type factor IX that is now out 3.5 to 4 years in the subjects who participated in that study with no late emergent safety signals. A phase 2b study that includes now this single point mutation on the original AMT 060 to make it into the Padua version. And this phase 2b study enrolled 3 subjects at the same dosing as the highest dose cohort from the AMT 060 trial. Now, if we go back to the AMT 060 results with the wild-type factor IX, there was a low-dose cohort, and then there was a higher-dose cohort. And then the lower-dose cohort at 5e12 vector genomes per kilogram, these were an older age group of individuals. They had pretty extensive rates of bleeding coming into the trial. The mean bleeds were about 14.4 per year, and their joint scores indicated that they had more advanced joint disease. They also accordingly were consuming more factor IX in units per year. The higher dose cohort, which was 2e13 vector genomes per kilogram, was a little bit younger population. These were median age of around 35. Their joints were in a little bit better condition overall. Their mean bleeds per year were about 4, and accordingly they used about half as much factor IX product per year. The phase 2b study that was dosed at the 2e13 dose in the 3 patients, these were most similar to the patients in the higher dose cohort from the AMT 060 study. The long-term expression now is impressive for the AMT 060 wild-type factor IX. In the lower dose cohort, the range is between just below 2% up to about 8.6%, and in the higher dose cohort, the mean activity has been maintained at 7.5% with a range of 4 to just under 11%. Notably, 8 of the 9 participants who had been using prophylaxis before they came into that study have now discontinued the prophylaxis, and they remain prophylaxis-free over the course of this long follow-up period. Now, what's exciting to observe from the phase 2b study, which now includes the Padua variant, we're now seeing mean factor IX activity at 1 year post-dosing of 41%. And that would have been predicted if we had the same transduction of the liver and the same expression characteristics from the AMT 060 vector, because the Padua variant in general conveys about a 6 to 7-fold boost in activity with the same level of expression. And the range for the 3 participants in the study was factor IX levels of between just over 30% and then into the normal range at just over 50%. So as might be expected with that degree of persistent factor IX activity, they've had remarkable reduction in their factor IX usage. All of the participants have been able to discontinue their routine prophylaxis. One participant did give a single infusion of factor IX for a suspected bleed, but it was later evaluated and determined not to be a bleed. From a safety perspective, AMT-060 has now given us up to 4 years of data on safety. There were some mild elevations in the ALT, which were asymptomatic in 3 of 10 patients. This did trigger a course of corticosteroids in those 3 individuals, but there was no loss of factor IX activity related to this. In the tranacogene dasaparvovec phase 2b studies, there was no clinically significant ALT or AST elevations to indicate any liver toxicity, and none of the participants required any immunosuppression. So overall, the conclusions are that the AMT-060 treatment was associated with long-term stable endogenous factor IX activity with no late emergent safety concerns, now over 4 years. This is the second longest follow-up reported in a gene therapy trial for hemophilia B. And then, excitingly, the single etranacogene dasaparvovec treatment has led now to a sustained factor IX activity in a functionally curative range with a mean factor IX activity at 52 weeks of 41%. No bleeds, no requirement for factor IX replacement therapy. Overall, this enhanced vector was well tolerated with no clinically significant elevations of liver enzymes or requirement for immunosuppression. So these data have supported the ongoing now phase 3, what's called the HOPE-B study. This is going to include 56 participants with hemophilia B, and this will give us data that will hopefully support this being able to be available widely for patients with severe or moderately severe hemophilia B.